Okay, so let's talk about Micro Lecture 1 quiz review. So it'll go over the topics that we would have discussed in weeks 1 and weeks 2, and we'll test over in week 3, okay? So chapter 1, we'll look at a brief history of microbiology, right? So there's a lot of scientists here, some classification of microbes that you're going to want to remember, right? So we first started to talk about, we talked about a lot of scientists, right? And so I won't talk about all the scientists, but I'll cover just some of the most important ones that you'll want to definitely hyper focus on, right? So Antony van Leeuwenhoek, right? This was that guy, right? He built his own microscope, right? He was an enthusiastic person. He looked at the images through microscope, and he initially called them animalcules because it was like tiny little animal things, right? Um, Looked at a piece of water, right? A sample of water, visualized these tiny animals, fungi, algae, single cell protozoa, called them animalcules. So he, was the first person really to see living microorganisms under a microscope. Okay. And so obviously we don't call them animalcules anymore. We call them microorganisms. But again, he was the first person to see these living microorganisms under a microscope. Okay. And so I'll get into some other scientists soon, right? So, how do we classify microbes? So, when we look at bacteria and archaea, these are unicellular, okay? So these organisms are only one cell. The whole organism is just one cell. And they also lack a nucleus. And so if they lack a nucleus, they're described as prokaryotes, right? Karyote coming from the word karyon, that means nucleus, right? Pro means before, right? So these organisms came before a nucleus, right? So that's why they lack a nucleus. So prokaryotes, these are organisms, again, bacteria and archaea, that lack a nucleus, okay? They're much smaller than eukaryotes. They're going to be found everywhere where there is sufficient moisture. Some are going to be in isolated extreme environments too. They're going to reproduce asexually. Also, they have a cell wall. So bacterial cell walls, they're going to contain this layer of peptidoglycan, or many layers of peptidoglycan, though there are a few bacteria that do not have cell walls. Archaeal cell walls, they're going to be composed of polymers other than peptidoglycan. And so peptidoglycan, if we just remember that, is basically, let's just break the word down, right? Peptido means peptide, glycan means sugar. So you have these sugar molecules that are interlinked or uh, interlocked together, and then you have these proteins or these peptides that cross-links them, right? And it holds these sugar layers together. And so that's the peptidoglycan layer, okay? And that makes up the cell wall of these prokaryotes. Now, fungi, which is another type of microorganism, these are eukaryotic microorganisms. So you got to remember that. These are eukaryotic microorganisms that have membrane-bound nucleus. So these eukaryotes, they have membrane-bound organelles, right? Such as a membrane-bound nucleus. So remember that nucleus, the house of the genetic information that, protects that genetic information, those nucleic acids, right? Such as DNA. It has two membranes around it, okay? So it's a double membranous organelle. Now, these fungi, again, they're eukaryotic. They differ from plants because they're going to obtain their food from other organisms, but they differ from animals because they also possess a cell wall. So our cells, we are animals, we don't have cell walls, right? But fungi and bacteria, archaea, right, even plant cells, they do have cell walls, right? Now, what's specific about fungi is that organisms are not photosynthetic. Okay, they are not photosynthetic. The types of fungi would be molds. These are multicellular fungi, right? Molds are multicellular organisms that grow as long filaments, and they can reproduce sexually or asexually. Unicellular fungi, we call it yeast, All right. And so these will also produce asexually by budding, and some can produce sexually. Okay. So remember, fungi, eukaryotic, not photosynthetic. They have a cell wall. We have unicellular fungi, which are yeast, multicellular fungi, which are modes. Protozoa are these microorganisms. These are single-celled eukaryotes. So it's only one cell, right? Single-cell eukaryotes that are generally motile. Got to remember that about them. They are generally motile. So they're going to be similar to animals in their nutrient needs and cellular structure. So they're going to live freely in water, and some will actually live in animal hosts. They can reproduce asexually for the most part, or even through sexual reproduction. Not all of them are photosynthetic, but most are going to be capable of locomotion. So they're going to have this ability to move. Whether they have a pseudopod is basically an extension of their cell that helps them to flow in the direction of travel. You can have cilia, right? Numerous short protrusions that help propel the organism through its environment. So the cilia kind of like beats in a sense, and it helps that organism move through the environment. Or the organism can have a flagellum or a flagella. These are extensions of the cell. Typically they're going to be fewer than a cilia. They're going to be much longer too than cilia. Okay, so if I was to draw, all right, so this would be like the cilia. Okay, that would be like the cilia, for example. But then this long thing, so these are cilia. This long thing right there, that would be an example of a flagella. Okay, much longer, much fewer, right, and much longer. Okay. Now here's the other scientist, right? So remember I had mentioned Antony van Leeuwenhoek, right? Built the microscope. He was the first person to see the microorganisms under a microscope. Initially he called them animalcules, but we now call them microorganisms. We also have other scientists, right? Ignace Semmelweis, right? So remember him, right? So he would have definitely developed, the hand washing type of scheme. But he wasn't really liked. We should say that, right? He was noticed, he noticed that women who were given or having babies in the hospital, they were more likely to die from giving birth than at home. So more women were dying in the hospital than women at home giving birth. And so he noticed that the medical students that were delivering the babies, they were doing autopsies ahead of time. So like in the morning, they were doing autopsies, and then afterwards they would give birth. And so he implemented essentially hand washing, right? So he required that hand washing must be done before delivering the babies. Obviously, people back then, they didn't like this, right? But he noticed that hand washing was super successful, right? And it decreased the amount of deaths of the women giving children. Unfortunately, he was unpopular, right? His ideas were unpopular, but they were successful. But, even with the success, he published the data, and the data was just simply ignored still, right? And it wasn't until Florence Nightingale, I'm going to skip over Joseph Lister for a second. It wasn't until Florence Nightingale came along, right? In like the 1850s or so. And she also essentially, we call her the mother of nursing, basically. And so she developed the antiseptic techniques in nursing and the standards of hygiene, right? So antiseptic techniques in nursing, standards of hygiene, also hand washing too, remember? You can put hand washing for her as well, because, she was a young girl, young British girl, good at math. There was a war going on at the time. She noticed that the soldiers from Britain, they were dying of like typhoid fever, right? So diarrheal diseases. And she noticed that more of these soldiers were dying because of the typhoid fever or illness rather than the war itself. So she implemented good hygiene practice, right? That's where we get right here, the good hygiene practice and hand washing, okay? And that's how she was able to save lives. So we also have Joseph Lister, right? And that's why you see that Listerine bottle right there for like antiseptic mouthwash purposes, right? So Joseph Lister, he was able to um, not develop, but really put importance to antiseptic techniques, and so he reduced infections during surgery. In the beginning, they used carbolic acid, which was an antiseptic, right? But it was very harsh, right? But it was there to prevent post-surgical infections, but it could provide even more infection or harm to the body because some of that carbolic acid, some of the residue would stay on the actual utensils sometimes. But nonetheless, remember, Joseph Lister is important for antiseptic techniques where he reduced infections during surgery. Okay, John Snow, right? Um, we can remember him about infection control and epidemiology, so we call him the father of epidemiology. He was able to track down the source of cholera outbreak in London, and he traced it to like a septic water tank. He basically used a map to find out where this cholera started at, and so he basically came up with the field of epidemiology. Right? So remember John Snow and epidemiology. Edward Jenner, in the 1790s, he was the first person credited with having the first vaccine. So he started the field of immunology. Okay. And I say the first person credited with it because remember, for smallpox, he created the vaccine for smallpox. He figured out if somebody had cowpox, it provided protection against smallpox. So we know the controversial story with Edward Jenner. Took the young boy, right, purposefully infected him with cowpox, right? The little boy got sick, and then he purposefully infected him with smallpox. But however, he noticed that the child didn't get sick when he was infected with the smallpox. So something with the body creating these antibodies gave him resistance against smallpox, right, from the cowpox infection. So that was the field of immunology, right? Edward Jenner. And of course, we have Paul Ehrlich, right, in the 1900s, early 1900s. He was trying to develop these magic bullets. Excuse me. These magic bullets, where he started the field of chemotherapy. So he essentially looked at chemicals that had selective toxicity. Selective toxicity being that they were harmful or it would destroy the pathogen, but it would be harmless, right? Non toxic to humans, right? Such as an antibiotic. Excuse me. Of course, we have to remember Hans Christian Graham. Hans Christian Graham developed the most widely used staining technique, which is Gram staining. So, Gram staining is going to be one of the first steps to identify a bacteria, right? That's Gram staining. One of the first steps to identify a bacteria. You take that culture, you try to see under a microscope, right? Is it gram positive or is it gram negative, right? And then you can do further tests. But again, gram staining is one of the first steps to identify a bacterium. Also understand other fields of microbiology such as serology. Serology is where we're basically studying blood, right? So we're studying like the antibodies in your blood, particularly using them as an indicator of infection. And so we have chemotherapy, like we just mentioned, development of and the use of drugs to treat infectious diseases, right? These chemical agents will harm or kill the microbe and be, harmless or non-toxic to humans. Bioremediation is another type of field of microbiology in the applied environmental microbiology field, where we use microbes to remove pollutants, right? Or you're using microbes to eat oil or use oil that if we have like an oil spill or something like that. Recombinant DNA technology or genetic engineering is where we alter microbial genes to synthesize useful products. So we're basically, putting a gene in a bacteria, making sure that bacteria produces that protein, for example. So an example would be insulin, right? So back then we got insulin from pigs. So now what we were able to do is understand, get the gene, right? The gene for insulin, And of course, insulin is a protein hormone, right? Specifically in humans, it's a protein hormone that's produced by the beta cells of the pancreas. But for persons that was not producing insulin, for example, like a type 1, or a person with type 1 diabetes, where they have an autoimmune destruction of their beta cells, so they're not producing insulin. In order to give them insulin, now we can get the gene that codes for insulin, put that gene into a bacteria, and have that bacteria read that gene and then make the actual protein from it. So we're making the insulin, right? So essentially we insert a gene of one organism into another for its expression, right? So we're altering the microbial genes to synthesize useful products. That's what DNA technology, or recombinant DNA technology, is in genetic engineering. So Robert Koch, right? Robert Koch, he developed these Koch postulates, which is just simply a systematic method to identify a causative agent to a disease. So he was able to identify that it was Bacillus anthracis, which is a rod-shaped endospore-forming bacteria that produced anthrax, right? So essentially what he did is he took, blood samples from a sick cow, and he found the bacterium anthrax, or Bacillus anthracis, was causing this, okay? He isolated the bacterium from the cow, grew it on a petri dish, and he inoculated it into a mouse. He noticed that the mouse died. He then got the bacteria from the mouse and identified that it was the same exact bacteria that caused the death of the mouse that caused the death of the cows. So what I want you to remember is Koch's postulates. And remember these in order, because you will be tested on these. Right? Number one, the suspected causative agent must be found in every case of the disease and must be absent from the healthy host. The agent must be isolated and grown outside of the host. So we're going to take it, right, outside of that diseased animal and grow it on a petri dish. When that agent is introduced into a healthy, susceptible host, the host must get the disease. So I take it from the petri dish, I put it into the mouse. The mouse now gets the disease, okay? And so the last part is the same agent must be found in the diseased experimental host, okay? So remember Koch's postulates. Also, we have Carolus Linnaeus. I got to remember this individual from the 1750s, actually, right? So he developed a taxonomy, right? Developed taxonomy, which was basically a system that classified organisms based on common characteristics, right? So essentially what it is he grouped organisms that can successfully interbreed into categories called species, okay? And he was also the person to develop the system of binomial nomenclature, okay? So when we understanding microscopy and looking at the microscopes, right, we have to understand the limits of resolution, right? The ability to resolve an object, or the ability to focus on too closely positioned objects, right? Being able to resolve that image, to see those images. Now, with the unaided eye, of course, we can see a chicken egg or a flea, right? Some large protozoa. But we do need a compound light microscope to look at like red blood cells, chloroplasts, bacteria that we look at in the lab, mitochondria, for example. But notice that we cannot see a virus, right? With that, see, it kind of comes like right there. You can kind of see it like that, right? We can't use the compound light microscope that we use in the lab to see a virus. It just won't work. So we have to use like an electron microscope, right? Where there's a transmission or a scan in an electron microscope, where we can see these super tiny molecules or organisms, right? Let me just say molecules. I'm not even going to call them organisms, right? Viruses, right? These parasitic agents, let's just call them that for now. We have to look at them through an electron microscope, okay? Cannot use a light microscope. Louis Pasteur, right? The father of modern day biology, or we should just call him the father of biology, of course, right? Of course, developed pasteurization, right? So in the 1880s, Louis Pasteur came about. He was the one to disprove spontaneous generation once and for all, right? Spallanzani, he disproved it, right? But it wasn't until Louis Pasteur he actually disproved spontaneous generation once and for all. Okay. So, like I mentioned, Lazzaro Spallanzani, he provided the evidence to disprove spontaneous generation. So we got to remember that. And that's because Needman failed to heat his vial sufficiently, right, to kill all the microbes, and he had not sealed them tight enough, right? So we understand that microorganisms existed in the air and it can contaminate the experiments. So spontaneous generation of microorganisms does not occur. All living organisms, all living things arise from other living things. Okay. And so, sorry, let me go back for a second just to stay on this slide. So, Spallanzani, he did provide evidence to disprove spontaneous generation. Now, Louis Pasteur, right, known for pasteurization, of course, um, and other things too, like the germ theory of the disease, things like that. But he used these bottleneck flasks, right? So now, don't judge me. Here's a regular flask, right? But, how should I do that? There we go. Look at that. This is a swan neck or a curved flask, right? The neck is curved, right? You can use your imagination for sure. But the flask is curved. And so he noticed that microbes could cause the broth to spoil. So microbes in the air could fall in here and cause spoilage, right, of the broth, right? Now, for this curved neck, right, microorganisms, they would actually fall, and I'm going to change the color for a second. They would get in here, right, because microorganisms are found on like the dust particles in the air, but they would drop right there, and they would stay right there. And, the fluid inside here, it wouldn't be cloudy. It would still be clear. So it was Pasteur who did this, and that's how he was able to disprove spontaneous generation once and for all. So Edward Buchner, another person, right? Edward Buchner, so Pasteur, Louis Pasteur, he understood that, yeast, for example, could ferment juice and create alcohol. Sorry, grape juice and create alcohol from that fermentation process. So initially, we thought it was microorganisms that promoted, fermentation. So we needed a microorganism for fermentation. However, it was Edward Buchner that demonstrated that fermentation does not require living cells. He showed that enzymes actually promoted those chemical reactions. And as a result, he began in the field of biochemistry. So remember that about Edward Buchner. So that was chapter 1, right? A lot of scientists, okay, a lot of classification of the microbes. So remember Antony van Leeuwenhoek pioneered the use of microscopes, first person to observe living microorganisms. He called them animalcules. You have Ignaz Semmelweis, right, introduced hand washing to prevent puerperal fever. Joseph Lister developed antiseptic techniques in surgery. Florence Nightingale applied antiseptic techniques in nursing, the mother of nursing. John Snow laid the groundwork for epidemiology. Edward Jenner developed the first vaccine, contributing to immunology. Paul Ehrlich developed the magic bullets for chemotherapy. And don't forget about Louis Pasteur, right? As well as Spallanzani, disproved spontaneous generation. We had Hans Christian Graham, developed Gram staining, a fundamental technique used for identifying bacteria. We will use Gram staining first before we do anything else typically. And then, who am I missing? Don't want to miss anybody. Just mentioned Edward Buchner, right? Field of biochemistry. Carolus Linnaeus, don't forget about Carolus Linnaeus, developed taxonomy, used binomial nomenclature. Joseph Lister, okay, just mentioned him, right? Trying to think of all the people so you won't forget. Koch, right? Robert Koch and the Koch's postulates established that criteria to link specific microbes to specific diseases. So remember all those scientists, right? I'm gonna mention some more too, though, but also remember like bacteria archaea. Remember, these are unicellular prokaryotic organisms that are smaller than eukaryotes and they have a cell wall containing peptidoglycan. And you have fungi, well, bacteria have peptidoglycan, archaea have something else other than the peptidoglycan. Fungi, eukaryotic, they obtain food from other organisms. They do possess a cell wall. They are not photosynthetic, right? Yeast are unicellular. Modes are multicellular. And then again, protozoa, these are single-celled eukaryotes. They are generally motile, right? Where they have like a pseudopod, cilia, or a flagella. Chapter 3 now, cell structure and function. So fimbriae are these sticky bristle-like projections, right? They are used by the bacteria to adhere to one another and to the substances to its environment. So it helps the bacteria to stick to one another and helps it to stick to its environment. These fimbriae, they're shorter than flagella, right? And they serve an important function in biofilms, right? Got to remember that. Okay. Now, so bacterial cell walls, we have gram-negative cell walls, right? Gram-negative cell walls have a thin layer of peptidoglycan. They also have a bilayer membrane outside of that peptidoglycan. So if I was to draw, sorry. Nope. So let me go back. Let me use my drawing skills again, right? So if I was to draw, a cell, right? So this is the inside down here, right? So we have the phospholipid bilayer. So you get the idea, right? This is a phospholipid bilayer. Obviously, we're going to have a thin layer of peptidoglycan. So remember, the peptidoglycan is like the NAG, NAM, NAG, NAM. You get the idea. And it's interlinked, honestly, via these NAG, NAM, NAG, NAM, right? So this right here, that's the glycan part, the sugar. You see how I'm connecting them now? This connection right here, these are the peptides, the peptide amino acids. That's why it's peptidoglycan, okay? So you have this phospholipid membrane, so the cell membrane. Again, this is the outside of the cell on this side. And you have the cell membrane. You have the peptidoglycan wall right here. And then on the outside of that, gram-negative cells, they have an additional cell membrane, right? This is the outer cell membrane, okay? That's what we mean by the outside of that peptidoglycan wall. So remember that about gram-negative cells. Again, they have a thin layer of peptidoglycan, but they have two membranes, basically, okay? And so they have what's known on this outer membrane, they have these lipopolysaccharides, okay? These lipopolysaccharides have a lipid portion to it, okay? And they have a polysaccharide portion to it. The lipid A portion, right? So this is highly stimulating to your immune system. So it will stimulate your immune system. And it can cause, fever, vasodilation, inflammation, shock, blood clotting. Okay. Whereas the polysaccharide portion, that's how we can identify the actual type of bacteria, right? So remember that about the lipid A portion, though. Okay. Now, because gram-negative cells have like that additional cell membrane, right? It may impede treatment of the disease. That second membrane benefits the bacteria, but it does impede our treatment of diseases from gram-negative bacteria. Now, gram-negative bacteria, they will appear pink, right? Pink from gram staining, okay? Gram-positive, I'm already going to start you off and mention that these will be, I don't have like a purple, but they'll be purple, right? Gram-positive purple because they have a relatively thick layer of peptidoglycan, okay? Also, they contain these unique chemicals called these teichoic acids and these lipoteichoic acids. Okay, so remember that about gram-positive bacteria. Again, they're going to appear purple. Right? They're going to appear purple. And up to, 60% of the mycolic acid in these acid-fast bacteria, right? They're going to be stained with, carbofusyl dye, right? And, in these mycolic acid, or I'm sorry, in these acid-fast bacteria, they have up to, right, I should say, 60% mycolic acid, right? And that mycolic acid is really important. It helps them to, their slow growth ability, and it also helps them from drying out or surviving desiccation, okay? So understand passive transports and the principles of diffusion. We know diffusion is just the movement of a molecule down its concentration gradient. It's going to go from an area of high concentration to an
Passive
area of
transport
low
includes
transportation.
diffusion.
We're going to pause that, right? Facilitated diffusion is essentially the same thing. We're just using a carrier protein, right? That helps to facilitate the movement. And again, osmosis is the movement of water down its concentration gradient, okay? So again, diffusion, movement of molecules down their electrochemical gradient through the phospholipid bilayer. So you have like small gases or lipid soluble molecules. So oxygen, carbon dioxide, steroids, things like that. Facilitated diffusion is you're still moving the molecule down their electrochemical gradient simply through a channel or a carrier protein, such as like a GLUT transporter, for example, for glucose. We have osmosis. Osmosis is the movement of water down its concentration gradient, okay? The cytosol, so remember the different parts of the cell. Cytosol is the liquid portion of the cytoplasm, mostly water, of course. And so in bacteria, that cytosol, or that cytoplasm region is going to contain the cell's DNA in a region called the nucleoid, right? The nucleoid is not the nucleus because it does not have a membrane around it, right? Remember, bacteria, they do not have membrane-bound organelles, okay? But in their cytosol, right, in their cytoplasm, they have these inclusions, right? These inclusions are basically these reserve, like little spots or packages within the cell, and it can be the sites of metabolic storage, right? So this is where the cell just has stuff at, right? It's able to store stuff, these inclusions, almost like these storage sacks. Basically, these storage sacks of stuff the bacteria need, right? So when we look at the effects of tonicity, right, remember if tonicity simply refers to the volume change of a cell. So if we're looking at the change to the volume of the cell, if I place a cell in an isotonic solution, that solution has the same amount of stuff. So water, again, water wants to move where there's stuff. So water wants to move where there's stuff. So it's going to move out and inside the cell because there's the same amount of stuff outside and inside the cell. Now, if we place the cell in a hypertonic solution, that means the solution has more than, right, the stuff that the cell does. And so water wants to move where there's stuff, so water will move out of the cell, and as a result, the cell will shrink and crenate and die, basically. If we place the cell in a hypotonic solution, the solution has less than the amount of stuff that the cell does, and so water wants to move where there's more stuff. So water wants to move where there's stuff. More stuff is inside the cell. So the cell will swell up, lyse, and burst. So hypo will lead to a hippo cell, okay, because it's going to burst. Also, we have to remember that glycocalyx, right, or the glycocalyx, right, however you want to say it, right, is a sugar coat, right? This is a sugar coat. It's a sticky, slimy coat, right? Um, it's going to be either a capsule or it could be a slime layer, basically. Okay, it could be a capsule or a slime layer. It's going to be present in some organisms, right? So it surrounds even some animal cells, like so, even eukaryotic cell, right? You look at the outside of your eukaryotic cell, you have a glycocalyx around it, right? So this is true for prokaryotic cells as well. So the capsule is going to be able to strongly adhere to the cell wall, and it helps the bacteria to like adhere. It also helps the bacteria to prevent, phagocytosis, right, from your body, right? It could even trigger endocytosis, right? So making the cell take it in, but it can escape phagocytosis, right, or being eaten. Okay, and then it's those capsule bacteria, really, I should mention that, that are really common. So the bacteria that have these capsules, these are those prevalent bacteria that actually cause disease or illness in us humans. Now the slime layer, for example, that's where glucose is going to be used in the glycocalyx for sure. But think of your teeth, right? So it creates that sticky plaque or the sticky coating, right, on your teeth, right? And then bacteria, they just feed off of that, right? But again, that glycocalyx is really important in helping to um, prevent phagocytosis and evade your immune system. Okay, so centrioles. Remember centrioles. So we talked about these before, centrioles and centrosomes. So centrioles. So I should say the centrosome first. Centrosomes are these regions, these organelles within the cytoplasm that's typically close to the nucleus. And this is where you find the centrioles at. The centrioles, these are those spindle fibers basically, so they play a huge role in mitosis, cytokinesis, and things like that. But they're not going to be found in all type of eukaryotic cells. So you're not going to find these, for example, in a neuron, right? So neurons are not able to undergo cell division. So if you lose it, you know, if you damage it, you're going to lose it essentially, right? So they're not found in all eukaryotic cells, but they're only found in eukaryotes, right? So you're not going to find these centrioles in a prokaryotic cell, right? The endoplasmic reticulum, which is a membranous structure that is, in a sense, continuous with the nuclear envelope, really, and it functions as a transport system, right, to transport these proteins as they are matured via post-translational modification. So the rough endoplasmic reticulum is important for protein synthesis because the rough ER has the ribosomes on it, and ribosomes are there to produce a protein, right? The smooth endoplasmic reticulum is going to be important for lipid synthesis, right? But remember, this endoplasmic reticulum is going to function as a transport system. Okay. Cilia, like we talked about before, are these short hair-like structures only found in eukaryotes. Okay, they're going to be shorter and more numerous than a flagella. Okay, and they're going to have this coordinating beat, right? So they're going to have this coordinated beating that's going to help propel the cell through their environment. Okay, also is used to move substances past the surface of the cell. Understand the roles of vesicular transport, movement within a vesicle. Endocytosis is essentially bringing something into the cell, right? If you're bringing something into the cell, you can bring it in such through phagocytosis, and this is the example right here. We're using phagocyte. Yes, phagocytosis is where you're bringing in like a larger object, for example, like a bacterium, and you're going to put it in a phagosome, right? And you're going to merge that phagosome with a lysosome because a lysosome have these degradative enzymes in it, and the lysosome is going to degrade the bacteria basically. And then, so that's, phagocytosis, bringing in like a large particle within the cell. You could also have penocytosis. Penocytosis is bringing in fluids, right, or liquids inside the cell. It's almost like cell drinking. You probably would have heard it before. And then we have exocytosis. Exocytosis is where you're dumping the contents out of the cell, right? So you're not bringing it in, but you're putting it out of the cell. So it's the opposite of endocytosis. So understand those concepts. Chapter 4, we're looking at microscopy, staining, and classification, right? So understand these metric units of length, right? You've got to remember that, right? So a deciliter is 10 to the negative 1. A centimeter is 10 to the negative 2. Remember the millimeter, 10 to the negative 3. Remember micrometer, 10 to the negative 6. Even nano, 10 to the negative 9th, right? So remember these, okay? Remember the parts of a microscope, right? You'll see this more in a lab exam, but you'll see, certain parts of it, or even on the lecture exam, right? So we have the ocular lens, right? It magnifies the image formed by these objective lens. These objective lenses are the primary lenses that magnify the specimen. And remember all the other parts though, but I'm going to focus just on those ones. Phase microscopy, right, is where we're using to examine living organisms, right? So we use phase microscopes to examine living organisms or specimens that would otherwise be damaged or altered by attaching them to a slide, okay? So there's two types, right? There's phase contrast microscopes and differential interference contrast microscope, right? For staining, right? These are some stains that are used for light microscope, right? We have simple stains. Simple stain is just we're using a single dye, one dye, such as crystal violet or methylene blue. And simple stains are there to reveal size, show us the morphology, so the structure or the physical characteristics, and like the arrangement of cells, right? That's why we're using a simple stain to see what does it look like, basically, right? We use differential stains, right? These differential stains are where we use two or more dyes, essentially to differentiate between cells or their structures. So Gram staining can differentiate between gram-positive and gram-negative cells, right? Which is typically, again, Gram staining, the first step in identifying a bacterium. We have acid-fast staining, right? That will distinguish the genre of Mycobacterium and Nocardia from other bacteria. And then we have endospore staining too, right? That will highlight the presence of endospores produced by Bacillus and Clostridium species. Okay, so remember these. Domain, so let's come back to our scientists for a second, right? So like in the 1970s or so, Carl Woese, compared these nucleotide sequences of the ribosomal RNA subunits. So he proposed that there was 3 domains, right? And these 3 domains are going to be determined by ribosomal nucleotide sequences. Now here are the 3 domains: eukarya, bacteria, and archaea. Very important note, viruses are not there because viruses lack ribosomal RNA. Okay, so viruses are not classified as living organisms. Okay, they're classified as intracellular obligate parasites. They're obligated to be inside the cell and they're parasitic in nature because they're going to feed off of the host. And unfortunately, the host dies or gets sick, right, for the most part. Okay? Again, taxonomy, remember who are we thinking about? You said it. Carolus Linnaeus. Okay? Developed taxonomy, right? That classification system. And it was the nomenclature, the binomial nomenclature naming system and identification. When we're looking at differential stains, right? Again, we're using more than one dye. We're using these differential stains to distinguish between different cells, chemicals, and their structures. Again, Gram staining, acid-fast, endospore stains. These are examples of differential stains, right? Using more than one dye to distinguish between cells. When we're looking at the parts of a dye, these dyes that are used as stains, they're generally salts. We have the chromophore portion, that's going to be the colored portion of the dye. Gotta remember, acidic dyes will stain alkaline structures or negative stains. Okay, so negative stain would be an example of a stain that's an acidic dye that stains alkaline structures. Basic dyes, they're going to stain acidic structures. Okay, Chapter 5, Microbial Metabolism. So, understanding that metabolism is the sum of all chemical reactions in the body, we have two major classes of metabolic reactions. We have catabolic pathways and anabolic pathways. Catabolic pathways, they release energy because they break larger molecules into smaller ones. So you're breaking down something. As opposed to anabolic, right, is where you're synthesizing something. So you're using energy to synthesize a larger molecule from smaller ones, right? So for example, if I was to make glycogen, right, so you make glucose, glucose plus glucose plus glucose plus glucose plus glucose plus glucose, right? And what does that give us? Glycogen. So I'm anabolically synthesizing smaller ones to make a larger molecule. Obviously, catabolic would be I'm breaking down this glycogen into many glucoses. Okay. There's different types of enzymes, right? And so these are a couple of different types of enzymes: hydrolases, isomerases, ligases, polymerases, lyases, oxidoreductases, and transferases, right? So you're breaking something with water, you're changing something around, right? The atoms around, you're gluing something together, you're breaking something apart, specifically not using water, you're transferring electrons, or you're transferring functional groups. Okay, so remember the different types of enzymes. Okay. Here's an example of an oxidoreductase type of enzymatic reaction, right? So oxidation is loss of an electron. Reduction is gain of an electron. This molecule right here was reduced because it gained an electron, right? It gained an electron. This molecule, do it in a different color. This molecule right here, it lost. See, it lost an electron. Oxidation is loss. Okay, so we understand that concept. Enzymes are types of proteins that go unchanged. Look at this. This is the enzyme. It's doing its job. It's doing its job. It's unchanged. Okay? It remains unchanged when it does its job. Okay? What's going to bind into the enzyme is a substrate. It's going to bind into the active site of the enzyme. Okay? Essentially, what enzymes do is, a lot of times they'll make or break covalent bonds in a controlled way. Okay? Now, there are some types of inhibitors that you've got to remember. Inhibitors are basically substances that block an enzyme's activity. We have competitive inhibitors. A competitive inhibitor is going to compete for the binding site. So it's going to bind to the binding site, and it does not allow for the substrate to bind. So it's competing, right? Competitively binds. A non-competitive or an allosteric inhibitor, right? Sorry. A non-competitive or allosteric inhibitor is going to bind somewhere else other than the active site. And as you can see, right, this is the perfect active site right here, right, that I'm looking at. But if that allosteric inhibitor binds right there, it changes the active site. So again, that normal substrate that will bind in there that needs this active site doesn't have that active site, so it can't bind in there no more. That's how you can inhibit something from binding. Either getting in its way, physically getting in its way and then blocking them and inhibiting them, or changing the shape of a molecule. Okay? So many protein enzymes are complete themselves. However, some are, these APO enzymes, these are inactive, right? If they're not bound to a non-protein cofactor. So some enzymes, right, they need a coenzyme or organic cofactor, right? Whether it's a metal or a solute or a vitamin, something like that, that will bind to the protein to then make it active. Understand cellular respiration. Glycolysis is the breakdown of glucose into pyruvate. So remember this image for sure. Glycolysis is the breakdown of glucose into pyruvate. If you do not have oxygen, no oxygen, you'll undergo fermentation, right? That pyruvic acid will get fermented and, you can make lactic acid, things like that. If you do have oxygen, this is now aerobic on this part. All right, this is aerobic. This is if you do have oxygen. Glucose gets broken down into pyruvate. Pyruvate then gets converted into acetyl-CoA. Acetyl-CoA the carbons of the acetyl-CoA goes through the citric acid cycle, right? And it's transferring. It's just really, we're just making different molecules. The citrate, the isocitrate, the succinyl, succinyl-CoA, the oxaloacetate, all of that, right? Essentially, what we're doing is we're making ATP, and we're making these electron carriers. We're making these electron carriers to take these electrons and drop them off at the electron transport chain. And that process, we are making ATP. And so in a eukaryotic organism, for example, right, oxygen is the final electron acceptor, right? Because oxygen has to accept that electron. And this is going to be an aerobic respiration. Okay? This anaerobic oxygen won't be the final electron acceptor. Okay? Chapter 6, we looked at microbial nutrition and growth. So when we look at microbial growth, we're talking about increase in the population, right? Not necessarily the increase in the size, right? So I'm not talking about here's a bacteria and then it goes like that. No, I'm talking about here's a bacteria, here's another one, 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 another one. So I'm talking about microbial growth in the form of increase in population, right? And that's going to be due to the reproduction of the individual microbes, right? As a result, we have these discrete colonies that we can see on like a petri dish. And we also have biofilms, right? So gotta understand this concept of biofilms. A biofilm is just simply a collection of microbes living on a surface in a complex community, right? So it's like different microbes. So think of your teeth, for example, right? Different microbes growing on your teeth that's forming that biofilm. So some organisms require oxygen. Some organisms, in the presence of oxygen, they'll die. So an obligate aerobe requires oxygen. So try to remember what this image will look like, right? In case you would to receive it. So obligate aerobes, you're going to find them closer to the top. So notice this top is a loosely fitting cap, right? So oxygen is like up here, but obviously it's high oxygen up here, low oxygen down there. So obligate aerobes, they require oxygen, so you're going to find them where the oxygen is at. Obligate anaerobes, these are organisms that are killed by oxygen. So oxygen is toxic to them. So obviously you're going to find them at the bottom where there's no oxygen at. Facultative anaerobes, these are organisms that grow better with oxygen, as you can see them up top mostly, but they can also live without oxygen. So you'll see them growing without, right? But you'll typically see more of them growing up here, okay? Aerotolerant anaerobes, they're unaffected by oxygen levels, so they'll be equally dispersed throughout, okay? Equally, because oxygen doesn't harm or kill them, harm or help them essentially, okay? Unaffected. So here's binary fission. This is how some prokaryotes would actually undergo reproduction. So essentially in binary fission, and you can watch that video too on your slides, the chromosome, the DNA will get replicated.
Most bacteria.
Right. The bacteria will get replicated. I should put it right here. The chromosome will get replicated. Then we introduce the septum or this cross wall right here. Okay. And then that cross wall is completed. And then you have two separately new, I guess, identical, right, organisms, right? And they just continue to divide. So again, this cell elongates. You're duplicating the DNA. You have that cross wall or that septum forming. That cross wall completes, separating the two cells essentially that have the exact identical information. Okay, you can see that septum right here, actually, that cross wall forming. Remember this graph. Okay, so the lag phase, right? This is where the bacteria. So this is a growth curve, right? You put your bacteria in a flask with broth, right? In a nutrient broth, for example, a liquid culture that you're growing. So I'm growing my bacteria. Should have did like that earlier, right? That's the fluid. You inoculate your bacteria in there. During the lag phase, right, you have slow growth, or essentially almost like no growth. In a lag phase, the bacteria is just getting used to the actual nutrients, right? They're just getting used to the nutrients. So they're just getting used to the environment too, right? So they're just growing a little bit slow, during the lag phase, okay? Then when they're used to the nutrients, right, they're going to grow quickly. So they're going to have an exponential growth. Exponential growth is where they're going to be utilizing all those nutrients, right? And so that splitting, that binary fission that we just talked about, it's going to be the quickest right here, right? So their generation time is going to be the quickest right here, okay, during that log phase because they're exponentially growing really fast. Then the stationary phase is, you see how we have this plateau, right? We have this plateau. So essentially at the stationary phase, you have the same amount of bacteria that are being born equaling the same amount of bacteria that are dying, right? That's why you have a stationary phase. So essentially, yes, there's still nutrients in there. The bacteria, they're using the nutrients, but as they undergo, respiration, right? Or metabolism, I should say, they're producing waste products. And those waste products start to build, right? That lactic acid, right, starts to build, changing the pH of this fluid. And remember, they only survive in a certain pH or a certain temperature, right? But if that lactic acid, right, acid is being, or the pH is decreasing, right, that's changing the conditions. And so some of them are dying. And then you reach this death phase where the nutrients are gone. Nutrients are gone. The waste products have now built up. And now you have a severe decline in death. So most of them, all of them are just dying now because there's no nutrients. There's too much waste there. Okay. So one way, so notice how, it's clear right here and then it's cloudy right here. Looking at and studying that turbidity, right, that cloudiness is one way that we can study microbial growth, right? So turbidity is an indirect method of studying microbial growth, okay? And that was it. Happy studies.